Okay, so we had looked at this dark and light photosynthetic reaction. So let me just briefly like take you through whole of this process, whatever we had done so far. And these are two slides which are really good kind of summary for all of this, whatever we had done so far. These basics, again, I don't think I need to revise at all. You know what is photosynthesis, definition, requirements, equation in general, importance of photosynthesis, that you are already aware. Autotrophs and heterotrophs, you know it very well. Just this might be the two new terminology, or you might have heard them before: photoautotrophs and chemoautotrophs. It's simply both of these are autotrophs. That means one is using photon or light energy, and the other one is using chemical energy or energy which is released from the chemical reaction. And then they synthesize their own food. These are the photoautotrophs and chemoautotrophs, which are the two types of autotrophs. And in heterotrophs, there are different types again. For example, what we call as parasitic or parasites, again this terminology you know. And there is something called as saprophytic. So parasitic is someone like an organism which takes nutrition from living organism, other living organism, that is parasitic. And saprophytic is the one of the organism which will take the nutrition from the dead organisms, dead or degrading or decaying organism. So that is saprophytic. So these are the two different types of heterotroph. So autotroph, you have photo and chemoautotroph, and heterotroph, you have parasitic and saprophytic. And again, saprophytic is the only new term probably which are dependent on the dead or decaying organism. And these raw materials, you know it very well, so I don't need to do anything there. So this is what we had seen. There are two types of reaction in the photosynthesis: light reaction, because it happens during the daytime, so it is light dependent. And there are different names: photochemical, because in this specific reaction there is this photon energy or light energy is being utilized for the breakdown of water and generation of ATP and NADPH. So that is what we had seen, and that particular reaction was called as photolysis. Right? Photo means because using the light energy, lysis of water, that is breakdown of water. So that is light reaction. And dark reaction is actually the Kelvin cycle, where whatever energy is generated in the light reaction, that is being utilized in the process of synthesis of glucose molecule. Not directly glucose, but like what we know of, that G3P, glyceraldehyde 3-phosphate is formed. From that glyceraldehyde 3-phosphate, glucose is formed. So again, you need not know the intermediates and stuff, but just to give you basic background, that's when I had introduced that C4 and C3 plant, because again, some of the questions, they are based on the C3 and C4. So there the confusion should not happen. We should know what is C3. It ultimately refers to the intermediate which is of 3 carbon, and in C4 plants the intermediate is formed of C4 type, that is 4 carbon compound, and that was oxaloacetate and malate. So that was the basic difference in the dark reaction. And it is also called as biosynthetic because it will synthesize certain biomolecules, or it is also called as biochemical reaction, because it will ultimately, through the biological process, it is synthesizing the chemical compound like glucose. So that is the brief about light and dark reaction as well. Steps, again, I am not going into detail. You can go back to the stages earlier, and there we have already explained this part. This I am skipping it, but just to give you a small idea about this. So what happens to glucose is further broken down into carbon dioxide and water. So it is synthesized from carbon dioxide and water. Again, it is broken down inside the cells into carbon dioxide and water. And that time 38 molecules of ATP they are generated. And that is when we say, no, glucose is a high source of energy. is because one molecule of glucose can generate 38 molecules of ATP, which is huge amount of energy. And that way, this is the part of or process of respiration. Again, this is not the respiration that what we call commonly as exhalation and inhalation. This is a cellular respiration. So at the cellular level, oxygen is taken and carbon dioxide is given. So that is a cellular respiration where the glucose is broken down and the energy is released. So this part, again, you should know. In some of the textbooks or even reference book, you might find 36 ATP, which is also right. But again, we need not go into those, why 36 or 38. I am leaving it at this point.
So just one question.
Yes.
This anabolic is a constructive process, like I synthetic process, right?
Yeah. So
So, and yeah, sorry, you were saying?
go ahead. Go ahead.
And there's this thing also called an anabolic steroid. So, are those the steroids that help you grow? Like, are those the things that you can
So
perform in?
So the word anabolic itself refers to synthesis, right? So whatever
Oh,
in the body, let's say we need proteins in our body. So from amino acid, proteins are getting synthesized. That is anabolic process. And when we need energy, proteins are broken down into smaller fragments, energy is generated. Or carbohydrates like glucose are broken down into smaller fragments and energy is generated. These are catabolic processes. And both of these processes, they are part of the metabolism. So metabolism is equal to catabolism, which is breakdown, plus anabolism, which is synthesis of molecule. So when we talk of metabolism, like in common sense, what we generally say is my basic metabolism rate is very high or very low, whichever way you want to say. So there people mainly intend to say the catabolic processes. They don't intend to say anabolic processes. But in
right.
general, in biology, metabolism involves catabolism as well as anabolism. High metabolism, that means the catabolic processes are more than the anabolic process. The high BMR, basic metabolism rate, that's what we call it. Okay, so again, I am skipping this part. We need not go into details of this. But yeah, it's good that that you know these points as well because they keep coming. Now, there are various experiments which are related to the photosynthesis. Oh no, sorry, we missed why this point went there. We need to do this first, leaf adaptations. You go down. Yeah. Yeah. So we'll first do the leaf adaptation part and then we will go to the experiment. So again, these are very common points. It's not rocket science at all. So what has been happening with the leaf so that it can adapt to the process of photosynthesis? First is larger surface area. Why does a leaf need larger surface area? Obviously to increase the amount of light that is being taken by the leaf. Now, if we think of a small chlorophyll pigment, one chloroplast will contain many chlorophyll pigment. One stack of grana will contain many thylakoids. One chloroplast will contain many grana, and within one one plant cell, there will be many chloroplasts. And then there are thousands of such cells. So you can imagine the amount of sunlight that will be taken by the chlorophyll molecule at the end. It's like growing higher in hierarchy. The number of ultimately the chlorophyll molecule within a leaf structure is too high, and they will harvest all the energy that is coming to the leaf structure from the sunlight. So that is increasing the leaf surface area will ultimately increase the photosynthesis. Leaf arrangement, mm, again, on a particular stem, the leaves, they are arranged in a such a way that they don't block the light of the other leaves. Now in dense bushy leaves, this may not be the case, but in most of the trees, the arrangement is such that they don't or they increase the absorption of the sunlight by making their arrangement suitable for covering whole of the area. cuticle and upper epidermis. Now again, presence of this cuticle will ultimately prevent the loss of water because it's a waxy material, and this we had seen in transpiration earlier. So the upper epidermis does not possess the chloroplast so that the light passes through them easily. It's a simple phenomenon. If the cuticle itself will have chloroplast, all the light will be there, will get absorbed by the chlorophyll pigment there itself, and it may not be able to pass through the different layers of the leaf. So that is when the cuticle does not have any what we call the leaf. The cuticle does not have the chloroplast as such. Then the numerous number of stomata. This is specifically required for more amount of carbon dioxide to be taken inside. So that is again one of the adaptation that leaves they do, and also that way they can control transpiration process also. More stomata, more control. So that way it is, of course, there can be more transpiration as well, but again depends on the temperature situation. But mostly it is more number of stomata, it is the better control over the transpiration process. Thinness of the leaves. The leaves they are like not so thick, and the reason for this is because the light has to penetrate through the different layers of the leaf. That is from upper epidermis to palisade mesophyll to the spongy mesophyll. So their thickness or thinness matters a lot. So thinner the leaves, better it is for the photosynthesis process. Again, this thinness of leaves is not observed in quite a few plants. For example, succulent plants or cactus variety. There you may not find thin leaves, right? So, but that falls in totally different category altogether. And the chloroplast itself, the leaf structure has lot of number of chloroplasts so that it can perform the photosynthesis, and that is one of the adaptation that plant has got over the time. Extensive vein system. Again, this is very much required for transportation of water through xylem as well as phloem. Otherwise, the food will keep on accumulating in the leaves itself if there would not have been phloem, or if there would not have been xylem, the water may not have gotten transported to the leaves part. So that way, extensive vein system is another adaptation by these leaves. Thin cuticle made of wax. Even though there is being the cuticle present on the leaf structure, it is very thin, and it also protects the leaf from any infection, and that way it does not allow the plant to get infected, and also prevent the water loss through without blocking the light. Otherwise, without blocking the light, I am sure you would have studied in the physics part what is optically transparent and what is optically non-transparent. So in simplest term, this cuticle, which is a waxy layer, is optically transparent. That means it will allow the light to pass through it without any change. Palisade cell layer at the top of the leaves. This is the one which is present close to the upper epidermis to absorb more and more light, and that way it can increase the photosynthesis rate as well. Spongy layer, again the air spaces within the spongy layer or spongy mesophyll area, it allows the gases to diffuse through the leaf. Again, this part we had seen in the transpiration process. And the palisade cells may contain chloroplasts. So whatever light is absorbed by these chloroplasts within the palisade cells, they can also do the photosynthesis function. So these are just altogether different structures which are present in the leaves as a part of adaptation to facilitate more and more of the photosynthesis. Any question? Okay. So if there are no questions, then the experiments. Now, so this one particular experiment is to prove that importance of light in photosynthesis. Now, this is a very simpler experiment. Without going into these details of the procedure, I can simply tell you that you take a leaf and keep it, not leaf, you take a plant, keep it in a dark room, so that the purpose of keeping a plant in a dark room is that all the starch that is present in the leaf will get utilized, and that is also called as de-starching treatment or de-starch plant, because in the dark room plant is not able to do the photosynthesis. So obviously starch whichever was already present in the leaf structure, that will be broken down into glucose that will be utilized for energy generation. So that is what is de-starching treatment. And this particular plant is again kept in light, and after few years the leaf is detached from the plant. I forgot to mention the black strips. So what you do is you attach this strip like thing on the plant, right, so that there is no light received by this particular area on the leaf, and then you keep the plant back in the light as it is, with the what we call as this black strip being there. Usually some people, they don't prefer black because it still absorbs some amount of heat, and that might cause but yeah, darker the color it's better, but as of now we'll stick to the black strip. So what happens is, because this area which is covered, it does not get light energy or sunlight, so it will not allow the photosynthesis to happen. Now how do we do this? We check it through a starch test. So what is the starch test? So there is a chemical reaction that happens between iodine and starch. So when we pour iodine into a starch containing substance, like which is present now in this case in our in the leaves, that will give certain color, right? So the region of the leaf which was under black strip turns yellowish brown, showing that no starch was made, and this yellowish brown color is of the iodine. Even after you add iodine to that, that strip or the area of the leaf below the strip does not change the color, and that is why there is no color change in this area. So that is what is shown here. While on the contrary, the area which was exposed to sunlight, which is outside the strip, it actually turns bluish black. So that is the complex of iodine and starch, which shows certain different coloration, that is bluish black. That means there is presence of starch. One of the steps that is done in this experiment is a boiling of the leaf for 2 to 3 minutes, and then again put it, not boiling of the leaf. The leaf is kept in the water, in the boiled water for 2 to 3 minutes, so that all the cells, they are dead, and then you put it in boiling alcohol, and then the chlorophyll which was present, which is the green color pigment, it will get removed from the leaf structure. So what you have in this leaf structure is everything without chlorophyll. Of course, small amount of chlorophyll remains, that's why the green color, but otherwise most of the chlorophyll is already taken out from the leaf structure, and the only remaining skeleton of the leaf, that is undergone for the starch test, and it shows that the area covered under black strip does not undergo photosynthesis, because there is no change in the color of this covered area, and the rest of the leaf, it shows color change to bluish black upon the iodine treatment. So that is the whole experiment. Any question again? Anyone? Okay, this de-starching process that I have already explained, I don't need to redo it. Simplest thing you need to remember why de-starching is required, because if we are doing a certain experiment which is talking about the survival of the leaf or survival of the plant, it is totally dependent on the photosynthesis. Now plant has a storage of starch. Even if you don't allow photosynthesis for a day or two, that plant can still survive because it has a storage of starch by then. So to deplete that starch completely, that is when the de-starching of a leaves is done by keeping a plant in dark room. So that is what is de-starching. Second experiment is importance of carbon dioxide in photosynthesis reaction. So in this case, again you take a plant and one of the leaf of this plant is inserted into a conical flask which has this potassium hydroxide. Again, remember we have to take a de-starch plant. That means this plant has already been kept in the dark room for time being so that the starch is depleted, and then you immediately take it for the experiment. So that is what is the meaning of this. So you take a conical flask, you insert the leaf into the conical flask, and you have kind of sealed leaf. So now sealed conical flask or sealed water, right? So that and within this conical flask or bottle there is this potassium hydroxide solution. Now the potassium hydroxide actually reacts to the carbon dioxide and it forms turbid solution, right? So that way, whatever carbon dioxide is present in this part of the leaves, this part of the bottle or conical flask, it will be already absorbed by the KOH. So there will be no carbon dioxide which will be received by this particular part of the leaf, that is which is inside the conical flask or the bottle. So again, we do the iodine test after a few hours. When we keep this plant in sunlight, half of the leaf will do photosynthesis, half of the leaf which is inside the conical flask will not do photosynthesis. You take out the leaf, put iodine solution on it. So the leaf which was exposed to the atmospheric air becomes blue back. Again, there will be, so in this region, so this part which is not exposed inside the conical flask or which is exposed outside to the atmospheric air, this will form not carbon dioxide, this will form glucose and ultimately starch. So this starch will react with iodine, it will show bluish black color. On the contrary, the part of the leaf which is inside this bottle will not have, wouldn't have done the photosynthesis process. So no glucose, so no starch. So no reaction with the iodine. So that way this part will remain yellowish brown. So that is the experiment which will prove that a particular leaf structure or a particular plant will always need carbon dioxide for the process of photosynthesis. So that is all about this. Carbon cycle is not there in the syllabus, so I am not doing it at all. Any question here in the importance of carbon dioxide? So there are only two of these experiments. There have been several questions being asked. Why do we need to de-starch? Why this strip is applied to this leaf structure, or why the leaves are boiled in alcohol, and so on, right? So you should be able to answer all of them. And even in this case, why is the KOH being used in the flask, or can any other molecule or any other solution be replaced with this, and so on. So this is very specific reaction of the KOH with the carbon dioxide. So that way this experiment is also specific to the importance of carbon dioxide in photosynthesis. Not really, the reactivity is not same. Though both of them they are bases, but the reactivity is not same. I mean, technically, yes, you should be able to use it, but then still the reactivity is not the same. Okay, with this, we conclude that this chapter is done.